Here we go again. Time for lecture number 4 on blood vessels. All right, I know you've enjoyed the first three. I'll try and make this one as entertaining, as fun for you as it is for me. So, let's get started. New topic, local blood flow. What I mean by the word local is blood flow to a particular organ or particular region. So not blood flow everywhere. I mean, obviously, if your heart pumps harder, cardiac output goes up, you're going to get more blood everywhere. What I'm talking about here is more blood in one particular spot. And your ability to increase, to augment local blood flow depends upon three things. Number one, the degree of vascularization. We can translate that into the quantity of blood vessels. Number two, the concentration of local regulatory factors. In other words, things made by active tissue that would influence and increase blood flow. Number three, total blood flow. I mean, this kind of makes sense. If my total blood flow, my total cardiac output goes up, my blood flow to individual organs, to individual regions is going to rise as well. All right, let's talk degree of vascularization. Your liver, incredibly active, high metabolic activity, thus it will have a high degree of vascularization, lots of blood vessels. Your Achilles tendon, on the other hand, aka your calcaneal tendon, well, it is not very metabolically active. I mean, it is made primarily of collagen. The fibrocytes that are in there, the cells that are in there, are kind of quiescent. So not very active, thus not a lot of blood vessels. And this also makes a lot of sense because having blood vessels within that tendon would mean less room for collagen, which would mean it would be weaker. Similarly, cardiac muscle, down here. Skeletal muscle, down here. You guys knew what those were. You saw the intercalated discs. You probably said intercalated discs when you saw this. And these guys are also very active. Hopefully, they're very active. And depending on their activity, they're going to have more blood vessels. All right, so let's say you decided to try and beat these 1970s era runners in a 5-kilometer race. You decide you want to train as hard as you could to see if you could run 5 kilometers as fast as Steve Prefontaine here. And he would run it in like 13 minutes, which is ridiculous. And you decide to train your butt off. This is your muscle biopsy before training. Little round things represent capillaries. This is your post-training. You grew blood vessels. In other words, you did angiogenesis, blood vessel making. Now, if you did not decide to do this, let's say you decide just to sit on the couch all the time, you would probably have some vascular regression where you would go backwards and you would lose blood vessels. That is not recommended. All right, what about vasoactive chemicals? So let's say you were competing in a triathlon, like Dave Scott here, and as his muscles are working hard, you can see his vastus medialis, his vastus lateralis there. As they work hard, their tissue temp goes up. CO2 levels, up. O2 levels, this is a tricky one here, down. Lactic acid levels, up. These are all triggers. These are all local things that activate the vessel. So the arterioles serving that tissue are going to vasodilate. Notice I gave you time to say it. Hopefully you said it. When you vasodilate, blood flow to the tissue will increase, thus dropping CO2, dropping lactic acid, removing heat, and bringing delicious oxygen. All right. Let's talk about local blood flow of a different sort. This is a inflamed leg. Look at that thing. Inflammation happens because of a rise in local blood flow. Anytime you got damaged tissues, they're going to do two things. Damaged tissues are going to activate white blood cells. Damaged tissues plus activated white blood cells. The combo are going to make inflammatory chemicals. Histamine is one we met before. New one here, bradykinin. Those guys are both local vasodilators. And the good thing about inflammation, I mean, not in the case, I mean, this is crazy inflammation. This is, ouch, inflammation. But the good thing about inflammation is the rise in local blood flow is going to deliver white blood cells, deliver immune proteins. Literally, inflammation translates as on fire because the area gets red, gets hot due to the increase in blood flow. All right, new topic. New topic is blood pressure. The force per unit area exerted upon the vessel wall by blood. All your vessels that have blood are going to have blood pressure. Pressure of blood is measured in millimeters of mercury, which is kind of weird, unless you were this dude right here and you were stabbing a glass tube into the carotid artery of a horse and you put a little mercury in there. The pressure in the artery of the horse would cause the mercury in the tube to rise. Then this dude could break out his trusty ruler, measure how high it rose, and that would be directly related to the pressure. Now, of course, you rarely do this in a clinical setting anymore. And by rarely, I mean never. And instead, you use a combination of a stethoscope and a sphygmomanometer, which you learned how to use in lab, hopefully. All right, explain this cartoon to me next time you see me. All right, you pass me in the hallway. Explain this cartoon to me. So let's talk about blood pressure. And we're going to start here with blood pressure in our arteries. And I know you already know, because you probably did this in lab already, by the time you're listening to this, blood pressure in your arteries varies. It goes up with every heartbeat, and then comes down. So with every heartbeat, every contraction, every systole, blood gets pushed into your arteries. More blood goes into your arteries than they can handle at once. So the arteries are forced to stretch. As they stretch, pressure rises. You're actually storing the pressure in here for while the heart is relaxing. And the peak value, which is 120 on my graph here, is known as the systolic blood pressure. Now, of course, once the heart's ventricle starts to relax, all right, blood, it's no longer pumping blood. The stretched arteries, because of their elastic tissue, their elastic fibers, they're going to recoil. As they recoil, blood is propelled onward. And as the blood leaves these elastic arteries, their pressure is going to drop. And so the blood pressure comes down until it reaches its nadir right here. And that value is called the diastolic blood pressure. So the low is the diastolic, the high is the systolic. Now, that's the arteries, okay? Like we got our aorta here. Blood pressure goes up and down, up and down. In our major arteries, same thing, up and down, up and down. In our arterioles, same thing, fluctuates. But notice by the time you get to the capillaries, no more fluctuation. Pressure overall is lower because you're farther from the heart. By the time you get into the veins, still no fluctuation. Pressure is far lower because you're even farther from the heart. And why is there no more fluctuation then? Why do we have fluctuation in the arteries, but not in the caps, not in the veins? The answer is elastic tissue. The arteries have the elastic tissue, caps and veins don't. Will this graph be on your next exam? Absolutely. All right, let's talk about what affects blood pressure. We're going to create a little model here. We've got a pump, the left ventricle. We've got our elastic arteries, and we have some arterioles. And what we can do is we can screw these screws in or screw them out and adjust the resistance, adjust the diameter of these arterioles. Well, let's figure stuff out. What would happen to blood pressure in these arteries if your ventricle pumped more blood? Well, heck, if you're going to pump more blood, these guys are going to get more blood, and blood pressure is going to go up. Thus, arterial pressure varies directly with cardiac output. Now, what if you tighten these screws here? Well, if you tighten these screws, if you narrowed your arterioles, it would be harder for blood to leave this area. More blood here, pressure goes up. Thus, arterial pressure varies with the resistance of your arterioles. Ooh, you're going to do this in class. This is a good one. All right. Whoops. Last but not least, what would happen to blood pressure in your arteries if you had more blood everywhere? Well, obviously, it would go up. So, arterial pressure varies directly with blood volume. And that, my friends, is where we'll stop right now.